Good afternoon, everyone. Thank you so much for joining us. We'll be getting started in just a couple minutes. Hello, everyone. Good evening or good afternoon, wherever you are. Thanks so much for joining us for Ask Me, our educational webinar series. My name is Audra Hines, Vice President of Care and Advocacy for the ALS Network. I want to thank our mission benefactor families, as always, as well as everyone who helps support our programming. The contributions from these individuals and families are absolutely amazing, and programs like this one tonight, as well as the multitude of services we provide, would not be possible without these generous donations. One of the ALS Network's key mission pillars is research. We fund and support the most promising peer-reviewed research and novel endeavors designed to accelerate the search for effective prevention strategies, treatments, and cures for ALS, and power progress in the neurodegenerative disease discovery space. To date, we've invested more than $19 million in ALS research efforts. The ALS Network, in collaboration with ALS United, has launched a public request for letters of intent as part of our rapidly expanding ALS Research Innovation Initiative. Promising submissions will be invited to submit a full proposal. And some examples of our recent research accomplishments include the ALS Network's Global Research Program launched a new scientific advisory committee in 2024. This is comprised of world-renowned researchers, scientists, and healthcare industry leaders. These ALS experts review and select the most promising projects and really moonshot efforts for funding. And this year in 2025, we're poised to launch our Community Research Committee, which is a group of people with lived ALS experience who will inform and shape our research priorities along with the scientific committee. Recently, we provided a grant award to AUTEX to help develop novel therapeutics for people living with ALS. And a generous grant was also awarded to Amprion, which seeks to understand the cause of sporadic ALS and actively assist in the development of potential therapies by supporting clinical and laboratory research. Last, I want to mention, you've heard this before on other Ask Me programs. We hosted our 15th annual ALS Research Summit, bringing together more than 200 world-renowned researchers, investigators, and clinicians, people in biotech and government representatives, and advocates in ALS and related fields, which was a wonderful program. We're so pleased tonight to have a special guest speaker. Dr. Michael McGrath is an emeritus professor in the departments of laboratory medicine, pathology, and medicine at the University of California, San Francisco. He's authored over 200 scientific articles in the fields of AIDS, cancer, and neurological disease since joining UCSF in 1985. And he has recurring NIH funding to support his research programs. Dr. McGrath has been a pioneer in the recognition of the role that inflammation and especially macrophages play in multiple diseases. His recent focus has been on the study of macrophage activation in the pathogenesis of ALS and other chronic diseases. He's also a co-founder of NuVivo Incorporated and is a member of our scientific advisory committee. So Dr. McGrath, I'm going to pass it over to you. Thank you so much for being here tonight.
I think I showed up a little early to the picture, but let me just start off. First of all, thank you very much for allowing me to speak to your group. I want to especially thank Sherry Stahl and Audra for this opportunity. It's coming at the end of, we had a meeting that NuVivo did with the FDA just last week to have a path forward for the drug I'm going to be talking about today. So let me just start off. And I guarantee you that the talk you're going to hear today is not something that you're going to be used to hearing. We're talking about preservation of vital capacity as a goal of ALS drug development. And I'm going to be talking about NP001 and how we've come to this idea of following vital capacity as an outcome for clinical trial evaluations of drugs. This is sort of a standard expression of ALS. Of course, we know that 30,000 people in the US currently have ALS, but the point that I want to make with this is down at the bottom, and that is almost half or more of ALS patients have respiratory failure. And so the question is this a movement disorder, a respiratory disorder, or as I'm going to present to you in a little bit, an immunologic disorder? And so I'm going to show you a little movie that actually gives you the punchline, and then I'm going to tell you how we got there. So let me just run this.
Amyotrophic lateral sclerosis, or ALS, is a progressive and uniformly fatal immunological disease affecting motor neurons and muscle and leading to respiratory failure. How is muscle function lost? Research has linked the development of ALS to increased activation of the innate immune system beginning in the periphery. From there, unabated innate immune dysfunction kills nerves and muscles important for respiratory function. Normally, the innate immune system acts as the first responder to threats, deploying cells to neutralize them. Once the threat is eliminated, the innate immune system rebalances itself by turning off that reaction. In ALS, the presence of disease-associated proteins prevents rebalancing, which leads to the destruction of nerve elements within the neuromuscular junction. With reduced nerve stimulation, respiratory muscles weaken, causing respiratory failure and death. As a prodrug, NP001 is the first innate immune therapy that facilitates the production of taurine chloramine, a key regulator of the innate immune system. Restoring innate immune balance in ALS patients may help control disease progression and promote survival.
So that's the punchline. That's all about the respiratory system as a target for not only drug development, but actually as a critical component of disease activity. But there's something that probably most of you don't think about very often, and that's the innate immune system. And what are we talking about there? Something pretty simple, but profoundly important to you through most of your life, and that is, this is just a typical, look at you cut yourself, and then there are macrophages, which are very central to the innate immune response, that recognize invasion, and they call help from the blood, and then you have granulocytes go into this area, and you fix the bleeding or the damage, and then you have resolution of that wound. And none of us are thinking that if we cut ourselves, we're going to die from it necessarily, and we always expect that they're going to heal. That's the component of the immune system that we call the innate immune system. It's happening at every minute of the day, in all of us, and it's under very tight control. If you think about macrophages, which I'm sure you don't, they're everywhere in the body, and they have different half-lives. And for example, in the brain, the macrophage in the brain is called a microglia, and it turns over every about 1% per year. Whereas in the blood, down below, the monocytes, which become short-lived tissue macrophages, live for about 3 days in the tissue. The other shortest-lived macrophages in the body are in the spleen. It's about 6 weeks. And so the blood monocytes are the cavalry, if you will, in responding to invaders into the body. But they're very short-lived. This is the component of the innate immune system that loses control, pretty much in evidence in the presence of chronic disease. So normally, inflammation and wound healing are balanced. And in almost all of us, we don't even think about it. But in chronic disease, you have too much inflammation and not enough feedback in turning this thing off. And so, depending upon where the inflammation is occurring, you have disease. And as you'll see, NP001 restores balance through a normal regulatory mechanism. And again, I would be remiss if I didn't show you a macrophage. This is a scanning electron micrograph of a macrophage. These are lymphocytes on top of it. It's a macro big eater. Normally it's phagocytic, and it's responsible for clearing the debris and infectious agents, and is our target for our discussion today. Very simplistically, the innate immune system is in balance normally from inflammation to regulation. You have to have inflammation to respond to an insult, and the regulatory macrophage balances this out. And in disease, as I said before, it becomes imbalanced. And so the critical molecule here, taurine chloramine, is made in the process of an immune inflammatory response and balances these things out. In chronic disease, there's not enough taurine chloramine in the right place at the right time. And NP001 is converted into taurine chloramine, augmenting the normal regulatory process. And so what you'd be able to show very easily is that NP001, which is a form of chlorite, is converted immediately almost in contact with macrophages into a long-lived molecule that's a chloramine inside the macrophage. So it regulates these cells in a long-term fashion. And if you just titrate out taurine chloramine, you can see that it downregulates, in this case, the y-axis is NF-kappa B activity. This is an inflammatory arm of the macrophage activation. And you see that there's downregulation and a dose response. And so you've got target engagement, which is taurine chloramine being converted from chlorite, and downregulation of inflammation. And so if you look at it from a cyclic point of view, you've got immune activation that makes hypochlorous acid, which of course would be bad in the tissues. It's like bleach, except it comes in contact with taurine, which is inside of phagocytic cells, and you make taurine chloramine, which causes regulation of nuclear factor kappa B, and then this regulates the immune response. And the problem with chronic disease we talked about is you have too much of this and not enough of this. And so what we see is that sodium chlorite is converted at the area of inflammation, by the way, where there's heme-associated iron that's available by the inflammatory cells. So it's basically a smart drug. It's inert until it comes in contact with inflammation. This now provides an extra boost of taurine chloramine, or in the case of our treatment program, a big boost of it to kickstart this balancing act back again, if you will. And so one of the things that I don't think that people appreciate very much is that there's a lot of work that's happened since 2020 through 2023, and I'm only going to show just the concept of it, and that ALS starts outside of the central nervous system, outside the spinal cord, at the neuromuscular junction. And some of the nicest work out of Stanford from Mike Snyder's group shows that the genes that they've mapped on a daily basis in the mouse model, the first ones that show up are at the dendritic processes of the neuromuscular junction. And what I'm showing over here is a motor neuron with the neuromuscular junction, and the classic approaches that have been taken by people for the last 20 years. There may have been close to 100 phase 2 and 3 clinical trials. There are 3 drugs that are approved right now, and I would argue that none of these are curing the disease. And so I'd say that fresh ideas are to be looked at critically. So with Mike Snyder's work and several other groups now focusing on the neuromuscular junction, we believe that ALS starts there as an innate immune response. And the proteins that have been identified as being in these neuromuscular junctions, things like TDP43 or SOD1, and the immune system ultimately recognizes them as being inappropriately present, and the innate response occurs. And again, this is from blood monocytes that turn into macrophages, and the approach is to regulate the immune response or regulate the innate immune system with NP001. So I'm sort of a simple guy. I'd like to look at simple readouts, and it turns out that if you look at one of the lower motor neurons, which gives rise to the phrenic nerve, that innervates the diaphragm, allows you to look at one objective measure rather than a lot of them at the ALS-FRS scoring system, to be able to look at whether your drug is having activity or not. And it's a critical one because what? It's directly related to survival. Breathing is important. And as people breathe less well, the quality of life goes down. And so the nice thing that we have available to us is the ability to do quantitative assessment of a breathing function through either measuring slow vital capacity or forced vital capacity. And so I would argue that going after this particular interface from an immunologic perspective is going to allow us to be able to treat ALS and to evaluate clinical effect of the drugs that are being evaluated themselves. So that gets us to the NP001 clinical trials. I should say that you probably haven't heard about NP001 for quite some time because there are 2 6-month phase 2 placebo-controlled trials done by Neuraltis. The first one ended in 2011, the second one in 2017. And so Nuvivo obtained all of this data once it became clear that vital capacity was an important outcome measure. And this was reported by, I think, really a seminal article from Jinsey Andrews in 2018, which really was the first to show, I think, really well that changes in slow vital capacity and forced vital capacity are related to survival, and that there were viable ways of measuring clinical benefit or clinical effects of drugs. So the clinical trials of 2 different phase 2 trials were done the same way, more or less. And the drugs were given intermittently. The first cycle was 5 days in a row. And it was, this is a 6-month study. And at the end of every month, ALS-FRS and vital capacity measurements were evaluated. And then biomarkers were also obtained. And again, these are older trials. At the end of both different trials, patients who had received 2 milligrams per kilogram, they're 115 total, or placebo is 110 total between the 2 trials. And so this is the program, intermittent treatment. And when we obtained all the data and looked at it, and this was reported in 2022, we did an evaluation of it as a post-hoc analysis. Of course, we didn't run these trials. And we noted there were 2 different things that appeared significant. Using the ALS functional rating scale, we were able to see that there was a subset of people who did not progress at all during the trial as compared to placebo. We're not going to be following up on that today. I reported on that at the ALS Summit. What we're talking about today is really the change in the vital capacity. Here you see change from baseline to vital capacity in this direction getting worse. And you can see the placebo loses about 2.5% of vital capacity per month. And this, by the way, is about the same as Jinsey Andrews reported. For patients less than the age of 65, they lose about 2.5% of their breathing function a month. And you can see the bifurcation here, the NP001 arm loses about half of that rate. And so the trial ends at 6 months. And so you go, well, wait a second, is there a prolonged activity? This seems unusual. Now, it's not unusual to think of it from an immunologic perspective. I just want you to remember one thing. We gave the drug at the beginning of a month, and we're measuring all of these things at the end of a month. So minimally, we're looking at changes that last for at least a month. And we had some data available related to one of the markers of inflammation called interleukin 18, where we looked at cells in vitro. These are macrophages from ALS patients who are treated with NP001. And this is now quantitative assessment of RNA from these different pro-inflammatory factors. And why am I showing you this? Because IL-18, it's an inflammasome-related factor that's disease associated with ALS, was measured. The IL-18 protein was measured in patients in the phase 2A trial, and an interesting observation was made. So if this, along this axis, is the IL-18 level in plasma, we see that in the 2 milligrams group, after a single cycle of drug, their IL-18 levels in the patients who received the 2 milligrams went down to the normal level and stayed there throughout the trial. Patients who received 1 milligram had it drop down initially, but then went back up, as if now we're showing a dose response or a threshold response, and that 2 milligrams per kilogram had a prolonged effect, whereas 1 milligram did not. So this then led us to think, well, wait a second, again, we've got evidence for activity against the forced vital capacity that appears to be prolonged. Same thing with this biomarker. Is there something else that we could measure to see how long the drug activity lasts? And so Bruce Forrest initiated at Novivo, initiated an intent to treat analysis of a survival study. Maybe some of you on this call helped us with it. It took about a year, but as you recall, there were 2 phase 2 6-month clinical trials that ended in 2011 and 17. The majority of these patients had passed away since participating in the trial, but we were able to identify clinical outcomes in 269 of 274 patients. So we had either dates of death or dates of still alive by September of 2022. So this gave us the ability to look at long-term survival. And again, we had it was intent to treat analysis. We looked at 2 different outcomes. The first was intent to treat, where everybody was looked at, and there was a hazard ratio of 0.76. There was an extension of life in these NP001 patients by about 4.8 months. And as we talked about earlier, patients less than the age of 65 were those identified by Jinsey Andrews as having the vital capacity loss of 15% over the 6 months. People over the age of 65, as we know, typically have a more sporadic outcome, mostly faster. And they have other reasons for inflammation than ALS. In this case, there was almost a year-long extension of survival. So then the question then is, how come these people are living longer? Is it really true, first of all? Are they living longer because of the drug? And if so, how could it be? So we did a finalist study, which is called a time to event analysis. In this case, at what time during a trial did they lose 15% of their vital capacity capability? In other words, if the median loss of vital capacity is 15% over 6 months, and we put a threshold of 15% on Could we determine whether there was a difference between placebo and the drug treated? And again, this was because the median loss of vital capacity rate less than 65-year-old patients was 15%. And so this is the time to event study we did. Event was defined as loss of 15% of respiratory function or death. The in vivo study was an intention to treat analysis of all patients. There was no pre-selection of people with inflammation or age or anything like that. So we looked at all patients. There were 114 evaluable, treated, 110 placebos, and the goal was to assess treated versus placebo in this time to event analysis. Now, what it would look like, if this is the change from baseline in vital capacity, here's 0, somebody who's not lost any vital capacity, 10%, 15%. And so we drew a line, and if somebody, and again, this is over 6 months, if somebody lost 15% at this clinic visit, over 15%, they would be scored as an event, and they would be lost to further follow-up. So this would be called an event. Just so you know, and the blue line, of course, had no event. The green had an event that was longer or occurred later than the original red one. And this is the result. So the result, if you look at it, is hazard ratio of 0.59, quite robust, with a 95% confidence interval of 0.39 to 0.89, p-value of 0.01. And what we're showing here is that the NP001 had fewer events, first of all, 37, compared to the placebo of 56, which meant that the placebos were losing their vital capacity at a faster rate than people treated with the NP001. And so this is sort of the take-home message that if people who are losing vital capacity at a slower rate than placebo, and by the way, this is a measure that's been used in the past and is quoted by the FDA for approval of drugs for treatment of idiopathic pulmonary fibrosis. They've approved 3 different drugs now, and that if you have a drug that changes this rate by 10%, that drug can get approved. And so this is the same kind of evaluation. Now, what we wanted to do as a final step was to say, if we achieve this end, and that is we kept people from losing 15% of the vital capacity, is that a survival advantage or not? Maybe it sounds obvious, but needed to be looked at. And so in this case, you see that patients who did not lose 15% vital capacity versus those who did lose 15% had an enormous advantage in survival. Over 2 years difference. Now, by comparison to placebo, there was a 12-month difference. And so the placebos also had an advantage if they didn't lose 15% of the vital capacity. But remember, there were far fewer placebos that were left because they had lost their event status during the trial. So let me just summarize here so I can get to the questions because I didn't want to get too complex a story across. Loss of respiratory vital capacity is a cause of death in most ALS patients and allows one an objective measurement of disease and drug activity. So why aren't we tracking this more often? Because it allows a very nice objective measure of the neuromuscular junction and how it's working in patients with ALS. The innate immune activation against misfolded proteins such as TDP43 or SOD1 may initiate ALS pathogenesis. Now, there are a variety of studies that showed that TDP43 can happen as long as it's present there and it's not being recognized by the innate immune system. It doesn't matter. But when it is recognized by the innate immune system, you now have got churning or chronic activation of the innate immune response, and therein lay the problem with damaging of the neuromuscular junction. We can regulate the innate immune system with NP001. Why is that? Because as we age, our ability to make taurine chloramine, and this is also part of the literature, goes down. And so this is a uniform ability to regulate the innate immune system that's really agnostic of what's causing the problem in the first place, whether it be TDP43, SOD1, anything that is continuously churning away at the innate immune system is doing so because most likely it can't regulate itself. We showed you intention to treat slowing of time to loss of 15% of the vital capacity. I showed you intent to treat extension of survival. And this is also correlated with the biomarker confirmation of long-lived regulatory activity. What we don't have is NFL data. These trials were done before NFL became a thing. And so I can't tell you about NFL results of this particular study. And I'm going to suggest that NP001 is the first immunotherapy targeting vital capacity as a measure of disease and drug activity in ALS. And I want to thank the group at NuVivo, who have been obviously incredibly supportive. Aria Zier and Vern Norval are our co-founders of the company. We've got a tremendous group of people who are driving the bus. I'll just leave you with one irreverent last finding. And I think this is electron micrograph. No, it's not. It's these guys are macrophages in your body, and they don't care what the evil is. They're going to pick it up unless they're dysfunctional. And so I'm just going to leave you with this as a thought and take any questions that you have.
Thank you so much, Dr. McGrath. This was really cool information, and I love that before we started, you said people are going to hear something they haven't heard before. So I always appreciate that. We've got some interesting questions. One gentleman says he has had PLS since 2004. Recently, his FBC dropped, and his neurologist recommended doing exercises to strengthen core muscles, and he's noticed some breathing improvement. Wondering if this drug, if you can answer, might be indicated for someone if you think about PLS and maybe based on some of his symptoms he's describing.
So the interesting thing is you gave us an advance. This is not a drug yet. This is an experimental molecule that we're in the midst of developing for ALS. And do the approach, so the approach is valid. I think that chronic inflammatory diseases, if you think about very few moving parts are happening in an innate immune response. And whether it be PLS, cardiovascular disease, Alzheimer's, Parkinson's, they all have commonalities that involve inflammatory macrophages and the inability to stop it, right? In other words, just stop it, will you? And so we have one version of how one can stop it, put it back under control. So that's a long-winded answer by saying it's not a drug yet, but I wouldn't bet against this approach.
Okay, and that's terrific to know. And someone else is asking if the participants had access to NIV throughout the trial. If so, was there any advantage noticed between those who did and didn't use NIV in conjunction with NP001?
I think that an excellent question. I think that we don't have that information, or NuVivo did not have that information. So I don't want to speak out of turn because I don't know the answer.
Okay, fair enough. Thank you. Are there any recruiting EAP or phase 3 trials for NP001 now?
There's not an EAP for NP001. We're in alignment with the FDA about the future development plans around a phase 3. So That's about all I can tell you about that right now.
Okay. And you mentioned, Dr. McGrath, at the beginning, a meeting with the FDA, and it might have prompted this question, asking if you've seen any slowdown in FDA responsiveness lately.
Well, we were worried about that,
Yeah.
but we had our meeting on at the beginning of the month. It was a virtual meeting. It wasn't a real meeting, but it was as real as an FDA meeting gets. They have their opinions and we have ours. But we're in alignment, I think, going forward. So it was a good thing.
That's great. I wanted to just sort of maybe a scientific, maybe a philosophical question for you. What gives you the most hope right now in the field of ALS research, knowing that there are only a few approved therapies right now for the disease? You're so plugged into the science behind all of this. What's giving you the most hope for our audience to hear from you?
Yeah, I think that's extremely important. I think that, see, I'm not a neurologist, hematologist and immunologist. So I look at things differently. We were the first to report in a very large study in 2005 that ALS was an inflammatory disease. I got stuff thrown at me by nice neurologists, but they still threw stuff at me. And it had persisted in the idea that as we age, the immune system doesn't work as well. I remember my first patient as a medical student had a pneumonia. He was a veteran and had we knew what the organism was. He was on the appropriate antibiotics and he died. And so I went to the resident and I said, "What happened? How come he died?" And the resident goes, "It's the aging immune system." And we are still at that level of sophistication about the aging immune system and what diseases are allowed to still happen. So I believe that the next wave of therapeutics is going to take into account things that happen with aging, because the immune system definitely ages, allowing all these things that we get, whether it be cancers or chronic diseases like cardiovascular disease, ALS earlier on, Alzheimer's later on. I think they're all going to have some relationship to the aging immune system. So I'm betting on immunology personally.
Thank you for that.
Immunologic approach. Yeah.
Okay. Yeah. And if people in our audience want to keep up with what's going on NP001 as you guys move forward, what's the easiest way to do that?
Well, we've published 6 papers in the last couple of years. If you just do ALS and McGrath, you'll
Okay.
see what we're doing. We just had another paper just accepted, and we'll have a couple more soon. So I think you'll keep up along the way, and there are press releases from NuVivo. I have to say that I'm as committed as anything. I'll just give you one other anecdote. I worked on AIDS at San Francisco General Hospital from 1985, arguably AIDS Central. And there was a type of AIDS called AIDS dementia. And there were hospices filled with AIDS dementia patients. Once antiretrovirals came along and stopped the disease activity and caused macrophages to cool off, people in the hospices, 60% went back to work. And that was the first time I said, you know what? All things that cause neurologic damage or neurologic symptoms aren't dead neurons. And so I believe that if you stop the insulting agents, whatever those might be, in the case of AIDS, it's HIV. And I started working on something I thought was going to be simple with Bob Miller called ALS. That was in 2001. It's not so simple,
No.
as we all know. But that's the point. I think that there's a lot that can be recovered, but you need to get the first stoppage of the disease before you can build it back up. And I'm arguing that we've got at least a toehold in the thought process of how to stop parts of it. So
That's encouraging. That's encouraging. A couple more questions have come in, and I'm sorry, I don't know what this one means, but you probably do. What do you think about Tregs?
no T-Regs.
Yeah.
Well, Tregs are part of the innate immune system as well.
Okay.
Uh, they they go away really fast. As early, I mean, Stanapel has been working on Tregs in ALS for a long time, and it's important work, no question about it. Um, uh, it's it's one component of the immune response that's it's not long lived enough, whereas macrophages live seemingly forever. So I'm dealing with a long-term evil. And I think the Tregs go way too fast for me to figure out how to help fix it. So I think that Tregs are important to study and see if we can, because they're part of the innate immune system, so I gotta like them. But I don't think we know how to control them and keep them around long enough yet.
Okay. Thank you. That's great. And last question, at least for now, that's in here. This person's asking to clarify: are you imagining NP001 as a potential treatment for respiratory function that's taken alongside a cocktail of ALS therapies?
100%. Yeah. I mean, this also treats ALS. It's a treatment for ALS. We just happen to be measuring vital capacity as a nice objective measure of disease activity. Whereas ALS-FRS, it's got things that it's got problems built into it. It's good. It's a good thing, but it's still noisy.
Fair enough. I don't see any more questions in here. I know we have a few slides to wrap up. Dr. McGrath, I really can't thank you enough for your time tonight and joining us. And we're always grateful for everybody in our community for your time and commitment and just for being here. If somebody could just pop up our last few slides, great, thank you. Once again, a big thanks to all of our sponsors for their support. And I just want to remind everybody, if you have other questions that were answered tonight, please do email us at askme@alsnetwork.org. The ALS Network brings our community together in various ways throughout the year to create help and hope for families impacted by ALS today and for those who will be diagnosed in the future. These are celebratory and meaningful gatherings that provide opportunities for connection and raise awareness and funds to benefit ALS Network's mission priorities. Please visit events.alsnetwork.org to learn how you can join us. And as you know, philanthropy fuels everything we do, and we're grateful to generous donors who make gifts of all sizes throughout the year, including contributions through an estate plan. To learn more about how you can support valuable programs like this one tonight, please visit our website at give.alsnetwork.org. And also watch for the short survey on tonight's presentation that you'll receive via email to help us plan for future educational programs. Thanks so much again, and we'll see you next time.